Will come up the final talk of this session. So it's Jannis Wollschlaeger from Reutlingen University and University of Hohenheim. Sorry. Welcome.
So, hello. Thank you for the opportunity to talk today. Maybe some of you knew me. I always was last year here, and now I'm happy to have new data based on that what I presented last year. So the topic is I have spheroids and printed them 3D via extrusion-based printing, and have there also a co-culture with fat cells and muscle cells. So the background we have in the first session, why do we need a 3D structure? Because we wanted to go further or more near to the native tissue of meat, and that's all just possible if we have 3D structures. And our approach is that to do that with bioprinting, and for that we need a bioink, and as you heard earlier today, that consists of biomaterials and also our target cells. So for cultured meat there are quite some different requirements for the biomaterials. So it should be edible, as you heard before, should be 3D printable, of course, in best case sustainable, have low regulatory hurdles for food application, and of course should be animal friendly. So we used, or I told you last year about what we use as biomaterials, and had there a kind of screening of different materials and figured out that gellan gum was the best that we can use for our cases, and that also mentioned fulfills the mentioned requirements I said before. And we tested that afterwards for their printing abilities, because the screening was without printing, and you can check that also at our latest publication I told you about last year. So what did we do now? We mixed fat cells and muscle cells with our gellan gum. In that case, bovine ASCs as primary cells and C2C12 myoblasts as a cell modeling for our muscle cells. And the problem with gellan gum is it doesn't support cell attachment. So our approach is that we just use the spheroids where the cells can attach to each other. So, and therefore we have two different scenarios. I will focus at the beginning at scenario 1, but it both starts with that we have our cells, let them form for 2 days spheroids in a shaking culture, and then do different things with that. And scenario 1, we just let them be spheroids in the shaking culture, and then let them differentiate either adipogenic for the bASCs or myogenic for the C2C12 cells. And after 14 days of differentiation, we put them together, mix them with gellan gum, and print them. And looking then how fine they are after that. So here are the shaking culture results. We see in the top line the C2C12 cells, and on the bottom the bASCs. Both are forming spheroids over the time, increasing their size. And now the interesting thing is, do they also differentiate over the time? And we can say, yes, they do. The C2C12 cells were stained with myosin, the red points here, and the bASCs storage lipid inside. So we see our differentiation was successful for both cell types, and with that we wanted to print them. On the top line we see they are still viable after the shaking culture compared to the dead control, and also after the printing process itself they are still viable. So we also checked now if there are still the characteristic for both, and stained them again in the gellan gum together, and see here with the red arrows the C2C12 spheroids, which have myosin, and the BASC spheroids, which have lipids inside. So we can say we have, after printing, maybe a ready-to-use product with our two components, fat and muscle. Compared to that, I come to scenario 2, where we have a different approach. We just let form our spheroids for 2 days, and afterwards we mix them together and print them and let them afterwards co-differentiate in our gellan gum. So for that, we need a co-differentiation medium. And first, we also checked, are they still viable over the time in co-culture? And yes, they are. So, but here we can't distinguish between C2, C12 and bASCs. So we also checked again how it's the differentiation going. And here we can see also differentiated myogenic C2, C12 cells and adipogenic differentiated bASCs within the same culture. Yeah. So, quick, I can come to the conclusion. So we can say we can differentiate both cell types in shaking culture, forming spheroids over 14 days. They're still alive, and also we can print them, and they're still alive after printing. Here, no co-culture medium is necessary. And if you can say, we have maybe a printing of a ready-to-use product after that. In scenario 2, we can mention also our cells still alive after printing and during the culture in gellan gum. And it is also possible to co-differentiate our bASCs and C2, C12 cells. Compared between scenario 1 and scenario 2, our spheroids are smaller in scenario 2. That's because we just take the spheroids after 2 days of culture in the shaking culture, and they're smaller and they don't grow or increase in size anymore after the printing. Also, we have to say we have to differentiate them after that. So all in all, we can say we have spheroids that can be used as building blocks for cultured meat, here in the example of bioprinting. So what are the next steps we wanted to see? Of course, I used here C2, C12 cells. That won't be the solution at the end. So we want to replace them with bovine muscle cells and have a co-culture for the same species. Also is to mention that we didn't use a complete serum-free medium here. So that's the next step also, where we can maybe switch that to a serum-free cell culture. And as you saw here, there are still many space between the spheroids. So it's also to increase the cell density inside of our bioinks to get more content of fat and muscle inside. And for that, we also figuring out in our group to increasing the cell mass itself via, for example, bioreactors, right? So I have to thank you for your attention, my team for supporting me, and also New Harvest for funding my work. Right.
So, do we have any questions? Mark has a question.
Nice progress. Congrats. Do you have a sense when you speak about increasing the cell density? Do you mean the cell density per spheroid in the spheroids, or the number of spheroids per milliliter of extrusion fluid?
Yeah, it's meant for the bioink itself, so that we get more spheroids itself inside of our bioink. Because in the shaking culture, we maybe could also increase the cell number, but it still works quite good at that point. And we don't want that the spheroids get too big because of cell death or something like that. And so the thought is more to get more spheroids inside of our bioink.
So for the extrusion, there's actually not really, that's not really an important parameter for the extrusion itself. You can work with any cell density in those spheroids.
That would be a thing we have to consider. Because we see if the spheroids are too big, we don't get them through the nozzle. So it's also a parameter we have to figure out and to see what is possible for us.
So there's a question over there.
Hi, thanks for your talk. I was just wondering if you were able to measure, or could you measure the efficiency of differentiation in your two processes? And then from that, would you be able to compare the yields of the differentiated cells between the two processes, the co-culture and the separated culture?
Right, so we didn't measure that yet. We maybe can do it via fatty acid analysis and protein analysis, but we have to do that in cooperation with the University of Hohenheim, but it's maybe could be a possibility to compare the products at the end, what maybe is more efficient for that, yeah, right.
Okay, no more questions. Then I thank you again for the talk.